Good afternoon. My name is Eleni, and I will be moderating this lecture today with my friend John. This broadcast is being carried out in a partnership with the Biological Control of Insects class from our entomology program, which is offered by Professor Angelo Palini and Madeleine Benzon. And in January, we are going to have more lectures presented here as part of this partnership, so stick around. Today, the theme is classical biological control. This is a topic of high relevance and that has been gaining space more and more in entomological studies. And you are having the chance to talk to an expert on the subject. So make sure your question is in the chat because it's going to be answered after the presentation, okay? So, as usual, today's broadcast will be divided into two moments. In the first moment, the guest will be freely addressing the subject in a presentation, right? And in the second moment, we will dedicate this space to the public, where the guest will answer the public's questions.
Welcome, my name is John Marulanda. My friend Eleni and I will be mediating this broadcast. Now we have the honor to recite the great Professor Simon Luke Elliot. He was with us in September, and with you, Professor, for being here again. Thank you. Professor Simon, or Sam, University of Amsterdam and Imperial College London. He is currently an associate professor at the Federal University of Viçosa and coordinator of the postgraduate program in entomology at UFV. Control of a pest, usually also exotic, aiming the permanent control of the pest. This biological control has been carried out with a over variety of target organisms, but most commonly against insects using parasitoids and predators, and occasionally pathogens. And today, Professor Sam is going to talk about three extremely relevant topics. Professor Sam, now is with you.
Is it my turn?
Yes, please.
Okay, thank you very much for the invitation. Thank you, Angelo and Madeleine, for accepting my invitation to talk on your course. Classical biological control is a subject dear to my heart. I worked on it for my PhD, but I studied it in my final year as an undergraduate, which I think I'll mention in a bit. And I thought I might talk about that today. Oops, let me just check something here. So I hope this is visible now. Classical biological control. So you've had a talk on conservation biological control. This is classical biological control. Just to repeat, Federal University of Viçosa, entomology postgraduate program. So this is where I work. Thanks to Insectum, a group of the student study group, for inviting me, and to the course here, which I put down here. Here's my email if anyone wants to talk to me about this afterwards. And support women in science. There we go. So this is a beetle attacking a bug. This is something we're going to talk about today. So I'm going to start with this example, and this example is a classic, hence classical biological control. So let's see, it's called cottony cushion scale. So it's a scale insect, which means it sucks the sap of plants. And it appeared in Southern California in 1868 as a new pest in a number of plants, but most importantly, citrus. It's prolificous, which means it eats a lot of plants. It can attack a lot of plants. And in English, we call it a soft scale insect, this particular group. I'll show you the group, Coccoidea. Here's a question, I don't know if it's visible, but why has Sam chosen such a lovely shade of pink? I hope you can see the pink, and we'll come back to this question in a bit. So, problem principally in citrus. Citrus is a major crop. Just as an aside, a friend of mine, and of Angela and Madeleine's, once fed me a sweet made from the flowers of citrus. Sweet from Iran. Very nice sweet. Anyway, so that's citrus and this problem in Southern California. And here is the insect. So these are adults, and these are the nymphs sucking the sap of citrus. Okay, this is its scientific name, Issaria puchasi, and it's a bug, Hemiptera, and it's a coccidae, or coccoidea is the superfamily. And the only means of control at the time, which is also the case with some other citrus pests, is complete destruction of the plants. There was no other way to control it. And so it was causing huge damage in citrus in Southern California, so in the 19th century. So the question then is, what can we do about this? And one thing is to think, well, where did this system come from? Where did the plant come from? Where does the insect come from? Well, you don't know where the insect comes from. So first off, think, where does the plant come from? Citrus comes from Asia, Australia, okay, Australasia. So this man here, Albert Koebler, decided to go to Australia in 1888. And I'm just putting here a copy from Anne Hayek's book, Natural Enemies. This is from 2004, but she told me she's got a new version out, 2018, with a colleague, Jürgen Eilenberg, so it's worth looking up. But this is a box from the book. It's worth having a read. I'm just going to summarize the story, classic story. And it's about the Vidalia beetle, it's called, because that used to be its scientific name. Now it's called Rhodolia cardinalis. And this guy went to Australia. He actually went to a conference which wasn't he wasn't supposed to look for natural enemies, but he pretended he was at the conference and went exploring. And he found two natural enemies that he thought were interesting. The one he actually thought was most interesting was this fly, which is a parasitoid fly. Here it's parasitizing. I guess this may have been an adult. So it's putting its eggs in here, and out will come an adult when it finishes its development. But they sent both sets of insects, so coccinellid ladybirds and this fly, back to California by ship. In 1889, 514 beetles were released. That's all they needed to release. Imagine that, 514 beetles. By the next year, they had control of the pest at a cost of $5,000 US. $5,000 US at the time was a lot more than now. But if you think about that, for 140 years of control of a pest that was destroying the crop, it's millions or billions of a return, financial return, basically by importing 514 beetles that spread, established, and controlled the pest. Just as a side point, you don't really have to do anything, nothing else has to be done except when people start spraying insecticides. When they start spraying insecticides and they kill the beetle, then you may have what's called a resurgence of the pest. You may have to introduce the beetle again, or the fly. They introduced 1,200 of the flies when it established as well. So both of these insects are established and controlling the insect. They're both specific. Well, this beetle is specific to this group, so it may affect other insects, but less so than a pesticide. But, you know, to think about the effects of this. So that's a classic case. That's probably the classic case, and is why it's called classical biological control. So what are the elements that we have here? We have an exotic crop. A lot of crops are exotic where they're grown, and they often do well where they're exotic because they have less of their natural pests and diseases. But then at some point, time catches up, and with globalization, which has been going on for hundreds of years, the pests that attack that, or the insects that are herbivorous in the native range of the crop, will arrive due to shipping, moving of, especially with citrus, for example, bringing cuttings or plants. They're often moved around. It's a big problem in citrus. And you have invasive pests. Invasive pests are a big problem, one of the main environmental problems these days. So you have this invasive pest that starts to destroy the crop. And one of the reasons why it destroys the crop is because it doesn't have its natural enemies. So the things that cause diseases or that predate it or that parasitize it are not there. So what you do, you look for the center of origin of the crop. So you think, where does, for example, coffee come from? If you've got a problem in coffee, it comes from Ethiopia and Kenya, Ethiopia and Kenya. So if you want to look for a natural enemy, I might mention this again if I remember, you look in Ethiopia and Kenya. You find natural enemies attacking, you find the insect, and then you find natural enemies that attack it. You select them. So you select them for attacking that insect. In this case, that's what I selected it for. And then you transport them and you release the natural enemies where there's a problem, and then you have control. So this is a very simple example. We're going to add more elements later. This is a simple example of a successful classical biological control program. It worked, and it's history, and it's classical. Saying that word a lot. There's also a healthy dose of luck. A lot of tests, a lot of tryouts don't work for one reason or another. In this case, there was a healthy dose of luck. It's often the case. And just to point out another term, a term we use often is a silver bullet. So the bullet that you put in a gun, and you have a silver one that will kill your werewolf. And so in this case, you have a silver bullet. So you have one solution to the problem, which is not very common, but it happens. So example 2 of classical biological control. And this is an example that's not so good. So here's our friend. This is called the cane toad. And it's called the cane toad because it's of interest for sugar cane. Okay, this is the biggest toad that there is. And what happened with production of sugarcane in islands in the Caribbean, also in Hawaii, and so on, with some pests. So in the case here, in a number of islands here, some of the islands down here in the Caribbean, there were beetle problems in sugarcane. And this beetle was found to eat, this toad was found to eat the beetles. So people introduced this toad to a number of these islands. One of the islands they introduced it to was the Puerto Rico. And it was successful, and this was in the early 20th century, so 1900 and something. So it was successful there, and so that was another interesting example of biological control, a classical biological control. So it solved the problem there. Here's the question again. Why has Sam chosen such a lovely shade of pink? Okay, so it worked here, and it was also introduced to another number of other islands, but in a lot of other islands, it didn't work as well. Then what happened? Well, they were growing sugarcane here in the tropical part of Australia, Queensland. And in 1935, because of the success in Puerto Rico, they introduced 102 toads from Hawaii. They had introduced it to Hawaii, and so they introduced them from Hawaii into Australia. And it became a major invasive pest. It became a problem. So here is its native range in blue, Northern South America and Central America and Southern Northern America. And here are some of the places it's been introduced and is a problem. Here, Papua New Guinea, islands up here. It's a big problem. And why is it a big problem? Because it's a generalist predator. There's an important lesson. Generalist predator. It eats anything it can find, basically. It's also a problem in Australia. This is a curiosity because it has a defense on its back. It produces hallucinogenic compounds. And so a lot of people in Australia lick the toad and they have a trip. They go on a trip, they go on a journey. I don't know if that's a problem, but it's very popular in Australia to lick toads. So let's look at how this program was done. Well, there was an exotic crop, sugarcane. Anyone know where sugarcane's from? There's a question. An invasive pest, so these beetles, maybe they weren't even invasive in this case. I'm not entirely sure. There was no exploration in the center of origin, no selection work, and there was no luck. It didn't control the pest and it became a problem. Just going back a bit, where is it native? Here. Sugarcane is native from here and from India. So that was not a success. And that was a major lesson in the 20th century. So those are a couple of examples, one of major success and one of major failure. So let's just pause for a moment for a bit of context. Let's think about different types of biological control, okay? I might return in January, if I can fix it, to talk about this, inundative biological control, in this case using microbial pesticides or biopesticides. And in this case, you're basically substituting a chemical insecticide or pesticide. And you're hoping for immediate control of the pest. So what you put in the field, you want immediate control. No second generation of your natural enemy. When you want control again, say even the same year, the same cycle in the same crop, you have to repeat it again. So if it's a fungus that you spray as a biopesticide, you spray it once, you may spray it again, and you may spray it again. Okay, so that's inundative. Inoculative biological control is a bit different because you don't put so many natural enemies in. You put some in and you expect them to grow, the population to grow. And what you hope is you have several generations of your pest, of your natural enemy, and it will become so populous that it will control the pest. It will grow faster, its population will grow faster than the pest. That's a summary. There are other sort of subtypes. One good example is predatory mind. So Angela works with that. Parasitoids are another example as well. And in this case, you're not expecting them to establish. So this is often used in protected crops, for example. And so next year, you have to do the same again. You have to release your natural enemies again. That's inoculative. So where does classical biological control come in? Well, it's the same first steps as inoculative control. You introduce your natural enemy, and at some point it will start to control your pest. But in this case, you're looking for permanent establishment of your natural enemy and permanent control of the pest. And you're looking for it to disperse if possible. You might have to help it with dispersal. But basically you're looking, especially if it has wings, that helps. You're looking for it to disperse. You may have to help it. So you're looking for a permanent solution. Now, if you're looking for a permanent solution, of course, you're not interested in companies. You're interested in governments. And that's a major factor, that you're interested in governments and not companies. So here you may have companies involved, and here it's mostly international collaboration, governmental schemes, and so on. So let's play again and think of some combinations of organisms. So I'm talking here about classical biological control of insects, and I'm going to focus insects here, invasive insects, and I'm going to focus here on on these cases, predatory arthropods and parasitoids, but let's stand back and look at other possibilities. Invasive plants are a huge problem here in Brazil, huge problem in Australia, Hawaii, a lot of islands. Vertebrates are a huge problem, invasive vertebrates, rats, cats, invertebrates, loads of invertebrates are a big problem. I put in marine invertebrates here. I don't know anything about control of marine invertebrates, but I know they're a big problem, taken around by ships when they carry water to weigh them down. When the ship has no cargo, and they'll bring water in, and then they'll go somewhere and they'll chuck the water out, and lots of starfish, jellyfish, lots of invertebrates. Microorganisms can also be invasives, okay? So coronavirus is an invasive microorganism, topical one. Natural enemies, so where can we think of biological control? Well, for plants, we can think of vertebrates, but these days we usually don't use vertebrates for biological control, okay? But you could think of it. You can think of herbivorous arthropods, often beetles, so insects. So this is a major area of classical biological control of plants, is using insects, often beetles. And you can also use fungi. So here at the Federal University of Viçosa, we have a colleague, Robert Barreto, in plant pathology, and he's been working on this for a number of years, very successful programs, and he's been involved in controlling major weed problems in Hawaii and Australia. Those are two areas where they're really strong in classical biological control. Only recently has there been use of fungi in Brazil for this, which is an interesting topic. It's not something I'm an expert on, but it's good that in Brazil this is starting to be used now, mostly by Robert Barreto and students he's worked with. And if you see the picture, the images, the results are fantastic. Vertebrates, well, I've put this here because people have introduced cats, for example, to control rats, but then the cats become a problem. So it happens, but it's not necessarily a good thing. There has been control of vertebrates using viruses. So a classic example is myxoma virus, used to control rabbits in Europe and in Australia. So rabbits were introduced, for example, European rabbit was introduced to Great Britain by the Romans, was introduced to Australia later on. They became problems. And in the 20th century, a virus was introduced, horrific virus, when you see the rabbits dying. But it's a case of classical biological control that worked. So here's the main subject here, invertebrates. So I've shown an example where vertebrates can be used. It's not a very good idea. And we're looking at some other examples. This is a mistake. Herbivorous arthropods. So sorry about that. But all of these can be used, okay? Marine invertebrates, like I said, I don't know. It would be interesting to have a look, think of possibilities. For example, I know there may well be viruses that could be of interest. Microorganisms, well, I'm thinking here in particular of pathogens of plants. And if we think of fungal pathogens of plants here, sorry, there are fungi that can be introduced to control them. So the example I mentioned at the beginning, with coffee, I started mentioning interesting project by colleagues, Robert Barreto again, is to control coffee rust. So he went with a colleague from the UK, Harry Evans, to Kenya and Ethiopia. I think they went to Kenya to look for fungi that attack coffee rust. And they brought to Viçosa about 100 or 200 different fungi, and they have some candidates that they plan to introduce to control coffee rust. So that's fungi to control a fungus. Fantastic, fantastic program. Viruses have also been used to control fungi successfully, especially fungi that attack trees. So there are viruses that can make fungi less virulent, less damaging. And that's another form of biological control, classical biological control. So this is a bit invisible, but this is a recent paper. It's not actually out yet. I had to ask Anne Hayek for a copy, and it's a summary of classical biological control introductions of entomopathogens and nematodes. And just to give you an idea here, she summarizes, or they summarize, that there have been 6,000 introductions of insect biological control agents against 588 pest species, a lot of them successful. 1,555 releases of almost 500 biological control agents for control of weeds. So these numbers are higher now in 2020. I think her book has a more recent summary. Insect pathogens, 164 introductions here. So that's just to give an idea of the different groups that I've just shown. So I've got two more examples I want to show you. Both are in Cassava. Both are very dear to my heart. And the first one is another classic. And this, it's hard to see, but this is the damage caused, and this is the insect. So this is a Mealybug attacking Cassava. And this is a classic case as well. And now we're going to understand why I've chosen such a lovely shade of pink here. So the Portuguese, during the slave trade, took cassava to Africa in the 16th century. It's from neotropics, of course, Amazonian region. Now, in 1973, in the Republic of the Congo and the Democratic Republic of the Congo, they were called Zaire and Congo at the time, this pest was accidentally introduced into these countries, which are on the west coast of Africa, southwest, and probably were taken by illegal movement of cuttings, so stakes that you use for planting. So there's movement of planting material is a big cause of these problems, of invasive problems. Now, 1971, 2 years before, the next case, the cassava green mite, was also introduced. So it's a double whammy, as we say in English, double catastrophe. But I'll come on to the cassava green mite in a minute. So 1973 was the introduction of this Mealybug. Sorry, just to show you here, Hemiptera pseudococcidae. Okay, that's the group. It's called Phenacoccus maniotin. This name helps tell you that it's highly specific to cassava. Pests of cassava are very often highly specific because of the latex and the cyanide. So there was this long search for natural enemies of this bug in Mealybug in North, Central, and Southern America, South America. But the Mealybug was not identified properly. It was misidentified. Why was it misidentified? Because the specimens that were collected were put in alcohol. And in the alcohol, they lost their pink coloration. And so it was misidentified. It was finally found in 1981 in Paraguay, just in the Paraguay basin, quite restricted range. So this is just to show you the importance of proper identification of your organisms. Just lost the color in the alcohol, and that lost about 20 years of research. So it was only found in this area in Paraguay in 1981. Later on, it was when I was working in Petrolina in the northeast of Brazil. It arrived in Petrolina, so in 1995, 5, it had got up to Pernambuco. It's not a great image here, but this parasitoid wasp was found in Paraguay, and it was chosen for release in Africa, and it was released by aeroplane, which is a good way to release a flying insect, especially, and it gave successful control of the Mealybug across Africa, so across the sub-Sahel and above southern Africa, a whole belt where cassava was grown. And of course, cassava is a hugely important crop, especially for resource-poor farmers who can't afford pesticides or anything. So This was, in fact, to date, it is in fact the most efficient foreign aid program ever. So the investment gave the most return of any international aid program that has ever been conducted. So even more efficient, more useful than the citrus program that I showed you. And just to show you some of the organizations, I said it's an international collaboration, and these are there are these international research stations here in Colombia, Cali, Center CIAT here, IITA in Nigeria. Embrapa was involved in 3 places, so in Petrolina, Cruz das Almas, and in Jaguariúna, São Paulo. Organizations in the UK and in Holland, in Amsterdam, were involved as well. So it was called Cabi Bioscience at the time. Now it's just Cabi, its name changes, but very important in classical biological control. And Imperial College was also involved, actually on the same site, the same location. That's actually where I did my PhD. And this is a paper that summarizes the story. It was produced in 1991. And I read this paper when I was at university studying biology. This was my last year at university. And I loved this paper, Biological Control of Cassava Pests in Africa. I absolutely loved this paper. One of the authors I met, Hans Herren, who's Swiss, very tall man. And that was when I graduated as a biologist. I then did my MSc, and it shows some of the extra steps, and these are the extra steps that you get nowadays in classical biological control programs. So you have your invasive pest, you have to identify it properly, you have to conduct the taxonomy properly. Exploration in the center of origin, you also have climate mapping. So look at the thermal requirements often. And then you're selecting your candidate natural enemies. We're going to come back to this question. I think I'm going over time already, but we'll see how we do. Taxonomy of the natural enemies is very important. So taxonomy is a field that is under threat, and here it's incredibly important. Climate matching of the candidate natural enemies is very important as well. Now, selection, usually you're selecting them to attack that pest, but you're also selecting an organism that has a certain degree of specificity. So not like the cane toad, more like the parasitoid. Parasitoids are great because they're usually quite specific. And that's part of your risk assessment. You see if it's going to attack other insects in this case. Yeah. Transport and release, well, usually another element is mass rearing. So to rear this wasp across Africa, it needed to release it, it needed to be mass reared. And that's different from the first example where it was just shipped. It took a month to arrive in California, but they managed to keep it alive on the boat. In this case, you have to look at its biology. And then once you introduce it, you have to monitor what goes on. You have to see if you get control, in part because you have to justify it to the people who gave the money. And you also have to do environmental impact assessments to make sure that you haven't caused natural, or to check if you're causing any other problems. Okay. So there's a lot of science nowadays in all of these steps, and these are big programs. So example 4. Whoops, we did it again. Here we go. So I mentioned the cassava green mite, lovely animal, tiny. And here it is on cassava. It's very specific to cassava. And it was introduced accidentally in Uganda, so in East Africa. It was first found there at least, and rapidly spread and was part of that catastrophe. It's a spider mite, but it's different from other spider mites. Spider mites are called spider mites because they produce web. The shade is pink, Angelo. I can see you asking in the chat. Pink, pink, pink. Look at this lovely shade of pink. But it's specific to, it doesn't produce web. It doesn't produce this silk web. It's different from a lot of other spider mites. So these were the organizations involved. And I forgot, of course, to mention, because of the success with the Cassava Mealybug program, the IITA built, by 1991, they had built this biological control center in Benin, which they speak French, as well as the local languages. So that's just to show one of the major impacts of that successful control program was this. So these were the organizations involved, and it was this organization that funded my PhD. When I got to Brazil and met the guy I'll show in a moment, Gilberto de Moraes, in 1994, they were still trying to release natural enemies from South America into Brazil. And they were spider mites, basically. So Steve Janinek was the guy working at IITA, and Gilberto de Moraes was at Embrapa, now he's at Exalc. They were introducing a lot of mites. Neozeulus was one genus, and Typhlodromulus is the other. And what they were looking for was high predation rates and high reproductive rates. So going back to inoculative control, the idea is that it reproduces more rapidly than the pest might. And high predation rate, so it brings the population down. And they introduced a lot of mites to Africa, but they didn't work. This is a terrible image, considering I did my PhD on this, but I was in the middle of nowhere in Bahia. So these are mummies of the fungus I worked on, Neozygites tanajoai, which is a fungus that kills the spider mites. And that was also introduced to Africa. Now, I produced this paper, 2008, which came from my PhD work, and it's on the failure of the fungus and this mite here to control the cassava green mite. Now, this is an important point in biological control. We have to be aware, we have to keep our feet on the floor and see if our natural enemy is actually controlling the pest. So these are data from the field cassava in Bahia. These are densities per 100 centimeters squared, so about the size of a cassava leaf. So this is up to 1,000 mites per leaf. That is a huge population on a leaf. Okay, and at the same time, I was finding, or a bit later, 120 predatory mites on this leaf. And that is also a huge population of predatory mites. And of course, this population crashed. Now, you might think this crashed because of the predators. I was also finding dead spider mites. So you might think this is because of the predators, because of the fungus. I did actually find a little bit of the fungus here, but it's not. What happened, if you look here, this is a third graph, and the scale here, this axis, is the proportion of leaves that were completely chlorotic. They turned yellow. And basically, all the leaves turned yellow and died and fell off. So what happened was there were so many spider mites, it killed all the leaves. And so that's why the populations crashed. So the predators are there, but they're doing absolutely nothing. And the fungus didn't appear. So this is just to show that sometimes they're not as useful as you might think. What was useful, however, was Typhlodromulus aripo, which is this predatory mite. So of the three species of mite, there were three species of natural enemy that established in Africa. This was the one that was successful. This is an interesting one because you find it in the growing tips of the plant, of the cassava plant. And it doesn't have a high predation rate, which is why they took so long to introduce it to Africa. Because it's not very good predator. Where it is good is it sits in the tip here of the plant, and when new leaves are formed, the cassava green mite, the females, come up to start laying eggs, but they get eaten, or their eggs get eaten by this low density of predatory mites that sit in the apex here. So that is not what everyone expected. So they did all these steps, all of this, released lots of natural enemies, and in the end, Steve Janinek was getting desperate. Something had to work because of the massive success with Cassava Mealybug. Something finally did work, but that was because of luck. And the reason why was found out later. It was found that it sits in the tip of the plant and attacks the mites as they come up, the pest. So that now has controlled the pest in Africa. So we're nearly there. Press stop soon. Just want to show just quickly show a few things. So this is another coccinellid ladybug or ladybird beetle, which has been widely used in biological control, started in the early 20th century, but it has become a huge problem. The Harlequin ladybird has a lot of different patterns. It's on every continent except Antarctica, and it's an invasive. It's a problem because it supplants the native ladybirds and reduces the diversity of natural enemies. Just to show you some other aspects. I haven't gone into microbial natural enemies, but this is just to show an example that was introduced into Australia from Israel, very successfully, a fungus that controls aphids. And going back to Anne Hayek's paper here, 164 release programs against 85 target pest species, 71% were established, 51% successful control. That's pretty good. Mostly Coleoptera and Hemiptera. Viruses, for some reason, highly successful. Viruses tend to be very specific. Finally, a paper that I published a couple of years ago with Harry Evans, I mentioned, and with Robert Bajertu from Plant Pathology. I haven't mentioned medical pests, but here's an idea that we could maybe look for fungi attacking Aedes aegypti, which is an invasive here. We could go to Senegal, that sort of area, which is the center of origin, look in the forests, and this is a plan. Let's see if it happens. And that's it. I finished. So thank you for the time. I think I was about 35 minutes. And that's me. I hope you enjoyed that.
Thank you, Professor Sam. Now we have two questions for you, and then we will open to the public's questions, okay? So the first one is, are generalist insects rarely considered for the classical biological control?
What was the word in the middle? What are generalist insects?
Because you talked about, for example, ladybugs. So I just thought about
the word.
Ladybugs generally
are generalist insects
not common.
No, you don't want generalists, not for classical biological control. You want specialists. So if it's pathogens, you want something that will only attack that species if possible. Insects, you want parasitoids. Most biological control with insects is done with parasitoids. Where it's predators, it's successful if they're quite specialized. Otherwise, you have problems with attacking other insects. Intraguild predation. The harlequin ladybird attacks plants. So it's a big problem. So you just avoid it. It's one of the first things. And it's the same with fungi, when you use fungi for biological control of plants, for example. So you do screening.
Mm-hmm. Okay. So the next.
Thank you, Sam, for your answer. The second question is, how long should we monitoring an organism that was released to the classical biological control?
What was the first bit? How long should we be monitoring an organism? That's a good question. Forever is the answer. That's not possible, of course. I think if you look at that paper if you really want an answer to that, I can't answer that, but there are now guidelines. So international organizations will produce guidelines for how long you should monitor. It depends on the organism as well. Some organisms are actually difficult to monitor. So I showed you Neozygites attacking Cassava green mite. Because there's already Neozygites in Africa, but different species, it's actually hard to monitor because you need to find them and do the molecular distinction between the isolates. So it needs investment, and who's going to pay for that? That's a big question. Certainly a few years, but then at some point, people stop because who's going to pay to continue, really?
Yes, that's a question that I always have been thinking about when we just introduce a new species, for example, in a place. So how long should we be monitoring this kind of species? So.
Well, you monitor. The thing with invasives also is it's very difficult to work out a priori what characteristics make an organism invasive. Similar to, you see that it's difficult to know what will be a successful biological control agent sometimes. You can say some won't because its host range is too specific, so you don't even try. But yeah.
Okay. So now we will move on to the questions asked by the public.
Which color pink?
The first question, the backstage. The question is from Mayara Picanso, and she's asking, when they began to pay attention to host range to select biological control agents? When they began?
I don't know really. Sometime in the, I think sometime in the around the 1980s and 90s, I think was when, well, the 70s, 80s, and 90s. If you look at that period, 70s, 80s, and 90s, you have a lot of research stations being set up across the world for this sort of work. For example, in Europe, the USDA set up a research station in France, in Montpellier, for this area. And at that point, there was a proper scientific approach being taken. And of course, people were aware of potential problems. So I don't actually know the answer to Mayada's question. I mean, it's something we could easily find looking at the literature, but I'd say it was probably around that time. I mean, I know it was a question when I first read about it in 1991.
Okay.
Thank you, Mayara, for your question, and thank you, Sam, for your...
Thank you, Mayara.
Mayara in Spanish. Agora, the second question of the public is Eduardo Constancin. He asked, hey Sam, very good lecture. Can I cite that the introduction of an exotic natural enemy to control an active pest is also classical biological control?
Yes. Thank you, Eduardo. Yes, you can. You can use an exotic to control a native pest. Yes. And you can use an exotic that you can create new associations. So you can find a natural enemy that has never encountered that organism. And if you think about most new associations don't work, but with testing in the laboratory, it can work. So yes, there are examples of that.
Thank you. The next question is from Bruno, right? So, when is a natural enemy considered ready to be used in classical biological control? Is there a minimum number of tests unknown target species that would be possible hosts, prey? Thank you for the question, Bruno.
Really, I think what happens is, I'm thinking about plants. I have plants in my head, but really, what you do is you'll take basic organisms that you conduct tests on for any intervention. So you'll look at bees and ladybirds and so on, and it can't attack those, obviously, and you'll try and find some relatives of the insect, say insect or mite that you're looking at. Sometimes people will introduce the things even without proper testing, just because they're trying anything, or that's been the history. And sometimes you may not have a large amount of those other organisms easy to rear. I don't think there's a definite answer to your question. So is there a cutoff point? I don't know. I'm afraid to must confess that the non-target aspect is not something I ever paid too much attention to, because I always worked on very specific organisms. Like Cassava green mite is only on cassava, and the fungus only attacks Cassava green mite. So, for example, with the fungus, you know that it doesn't attack Tetranicus urticae or other spider mites. You already know that so it's not going to attack anything else. So that's an easy answer. We don't really, then you test it you do stupid tests against chickens and against bees and so on, and you find that it does nothing, which is what you expect. Really. They're not stupid, you have to do them, but you know the outcome. And I think it's the same with a lot of parasitoids, because you already know that they're going to be quite specific, and you look at some close relatives, and if they're not attacking close relatives, they're not going to be attacking other insects.
Thank you, Bruno, for your question, and thank you, Sam, for your answer. The next question is of Andre. He asks, is there a case of success or intent of classical biological control of a species that was threatening directly a natural enemy of a pest? For example, some hyperparasitoids.
Ooh, I don't know, we're getting complicated now. I can't think of any. The only one I'm trying to think of is if there's been an effort with the Harlequin Ladybird, because that would be an obvious target. And we know that there are, I think, 100 parasitoids that can attack it. So I think there's been research looking at potential introductions of parasitoids to control the Harlequin Ladybird. There's also some fungi that attack it. There's a very interesting fungus that attacks it that is sexually transmitted in the ladybirds. So that's the only case I can think of. But that's partly because I can't think of many cases of the second trophic level being the problem. Yeah.
Okay, so thank you. The next question is from Angelo Calini. Do you know how is being the activities of the Brazilian quarantine in Jaguariúna, São Paulo, for projects on classical biological control?
No, but I can tell you a story. In 1996, when I met Angelo in Jaguariúna, I was at Embrapa, Jaguariúna, near Piracicaba, I was working with Gilberto de Moraes. I was at the end of my PhD. And they were setting up the quarantine station, so in 1996. So that was a big effort by Gilberto de Moraes, who's now retired, but at that time had moved to Exalc. And good God, I've forgotten her name, Betch. She died of cancer, I'm afraid. She was working on that. And Luiz Alexandre Nogueira de Sá, who died last year. So they were the people who set up the quarantine in Jaguariúna. So it was set of not glass houses, because it was a polycarbonate. And I was trying to help set up this quarantine facility. And so you're supposed to be have all these ways of entering the quarantine facility, and I was looking at. I was supposed to translate the instruction manual for the pump from Japanese into Portuguese. I was trying to help do that. So it was all at a very early stage, and it subsequently got set up and is now one of the quarantine places for bringing in natural enemies in Brazil. I don't know the current situation. I met Luiz Alexandre in Gramado a couple of years ago. All I know is that it has been working since then. When I showed that map with Kabi Bioscience and Amsterdam, those places, and also France, one of the reasons for their involvement is the possibility of taking tropical natural enemies to a temperate climate and a quarantine facility. So I worked in a quarantine facility there in Kabi Bioscience. And if it escapes, it's in a temperate environment, so it won't be a problem. I'm not answering Angela's question at all, because I don't know the answer. But I'm just saying, just since he mentioned that facility, it's very important to have these facilities. So in the UK, there's this one where I worked in London. Well, we'd have to find out. I have to look on the website. I'm not very up to date on this in Brazil.
It's okay. Thank you, Angelo, for your question. Thank you very much, Sam, for your answer. The next question is of Madeleine. She asked, Sam, do you think the Convention of Biological Diversity limits biological control?
Well, what I've read is that there are expected to be less classical biological control programs in the future because of stricter legislation. But because of the stricter legislation and the tighter controls, so it's more controlled these days, there are going to be more successes than before. That's the opinion I've read. I think the interesting thing on that aspect about biological diversity is that the areas which have the most characteristic fauna and flora, or the nations or the places that are the ones that have invested most in biological control, in classical biological control. And that's largely to protect their flora and fauna that are threatened by exotics. So two of the places that are most important are Hawaii and Australia. Incredibly strong on introducing natural enemies for biological control. And they're the ones who have the most to lose by introducing natural enemies if it goes wrong. There's a cost benefit ratio, and so risk assessments are done. I think there's less biopiracy nowadays than there used to be. So that example of taking natural enemies from California, from Australia to California, that was biopiracy. course, successful biopiracy. I don't know if that answers Madeleine's question, but I think the stronger controls are going to make, also, you don't want things to go wrong, because when it goes wrong, it's terrible. You see that nowadays, the UK, 10 years ago, started introducing, had its first classical biological control program, having worked on the UK being incredibly strong in the area, but its first introduction was 10 years ago. Brazil has its first introduction of fungi now, recently, despite a lot of work in Brazil on classical biological control. It's very, the diversity, or the biodiversity in Brazil is very important. So it's not a direct answer, but might help.
Okay, thank you, Sam and Madeleine, for the question. And I think we have the last question, and that is from Tadeu Souza. Sam, is there an example of a microorganism that has been improved to be more virulent for use in biological control?
I don't think in classical biological control. Because the thing with classical biological control is you may not want high virulence, because what you really want is establishment and persistence. And then, yes, you want it to kill the pest, but if it kills them too quickly, it may not give you good control. So work has shown that, for example, with spider mites, the best biological control agent for a spider mite may, in a patch of spider mites, a group of spider mites, can be a natural enemy that is not very virulent, because it interacts with that patch for longer and produces more natural enemy propagules that then go to other patches. So that's work that was done in Amsterdam while I was there by a guy called Bas Pels, who showed that with predatory mites. And you expect the same with microorganisms. If they control a local population, if they're very virulent, they might not produce many propagules that then persist or control other patches, other populations. Where you get a lot of interest in improvement, in quotes, of microbiological natural enemies is with biopesticides. And people try and increase the virulence for two reasons. One, if you increase the virulence, then your microbial biopesticide may not persist in the environment. It may kill the insect too quickly to produce any propagules. So that can be good because you're reducing the persistence, like you reduce the persistence. You don't want high persistence always of a chemical insecticide. And then the other reason is to increase its efficacy. And so there's this study that Kemeny showed in the seminar last week, increasing the virulence of metarhizium for control of mosquitoes. But again, that's as a biopesticide. I was very skeptical of that, but they've shown that it gives better, longer control. So I hope that answers your question, Tadeu. For classical biological control, I don't think so. I think you can have choice of strains. You can choose a more virulent strain, but then you ask, is virulence what you want? Or do you want persistence? My whole PhD was looking for how this fungus persists in the field, looking for resting spores, which I found in my last year.
Okay, thank you, Sam and Tadeu, for the question. So I think there is no more questions here. So if you wanna say something else, just to finish the.
No, I just to highlight what I find most interesting of this area and some other areas of biological control or of pest management is the importance of international collaborations. You see it also with, for example, sterile insect technique or genetic modification for control of insects like Aedes or use of Wolbachia. And these are non-profit initiatives. And that's where I think it gets, I mean, it's interesting when you get biopesticides making money, which we're starting to see, which is good, and use of natural enemy, other uses. But it's also very interesting when you get these international collaborations, and those are things that we need to emphasize. And it's very important for classical biological control. And just to say again, that it's a very, very efficient way of investing money. Can be very important. Okay, that's all. Thank you.
Okay, that's all. So we thank you, Professor Sam, for your brilliant participation.
Thank you, John.
And now we are ending this afternoon broadcast. We inform that this video will be available on our YouTube channel. And do not forget to subscribe to the attendance list on the link in this video description. Okay? Yeah, John?
Obrigado.
We thank you for attending this event. Our lecture cycle will continue in January. Time, date, and time are to be defined. You are welcome to enjoy us, and you can get a certificate. Once again, thank you very much, Dr. Sam, and all staff participate of this broadcast. Good afternoon, everyone. Take care, and my best wishes.
Thank you. Thank you, Kemini.
Bye-bye.
Bye-bye. Merry Christmas.